Hold on. Okay, so thank you for coming to Department of Life Science International Women's Day lecture, and I hope you can hear me well. And this is our 6th year Department of Life Science International Women's Day lecture series. And if you're interested in previous lectures, please go to our website. And if you just Google search for DoLS, Department of Life Science and International Women's Day lecture, our page will come on the top. And today's lecture will be also recorded and uploaded onto our website. So I am delighted to introduce today's speaker, Professor Joy Burchell. After completing her bachelor's degrees in biochemistry at the University of Sussex, Professor Joy Burchill went on to work for the Imperial Research Fund, now known as Cancer Research UK, and she undertook a PhD, postdoc, and the position as a senior scientist there before joining King's College London in 2007 as a professor in glyconcology. She devoted her career in investigating the relationship of glycosylation and breast cancer, and she opened up the door to a completely new field of research and contributed to the development of personalized therapy for breast cancer. So over to you, Joy.
Thank you. So first of all, I'd really like to thank Kenji for inviting me to give this talk on International Women's Day. I've been asked to give you an idea of my career journey, and I want to discuss this, and I thought it would be most interesting for you to listen to discuss this within my research. So you'll see how my research and my career pathway evolved. The picture here is of King's College London on the Life Science and Medicine campus looking across from the river, and this is obviously the Shard. This is Guy's Hospital, and the Life Science and Medicine campus is sort of tucked in behind there. So that's where I am at the moment. So unlike some scientists, I had really had no idea what I wanted to do when I was at school. This was in the late 60s and early 70s, and I even took RE as a A-level because I really had no idea what I wanted to do. And I thought this would be very good if I wanted to teach, because really in the early 70s, teaching was one of the sort of expected career pathways for women. However, I really enjoyed biology and chemistry, and therefore I took biochemistry at college. But still, I didn't really know what I wanted to do. But when I graduated, I was given this booklet that said, scientists who are employed by biologists who are employed by various institutions. But before I go on to tell you a bit about that, I just wanted to say that although I didn't really know what I was going to do with my career, my personal life seemed to be very well organised, because I actually met my husband at 16 and we were married at 20, and we've been very happily married ever since. We were married at 20, so I was 20, he was a little bit older. So on the next slide, it shows the Imperial Cancer Research Fund as it was when I first went to work there. So I had this little booklet that said institutes that employ biologists. And so I wrote to the Imperial Cancer Research Fund asking for a job. And I was given a job there working with in the Department of Androgen Physiology, which was run by Ian Mannering, and I was working with Steve Higgins. And this next bit here just shows you an insert. This is what's the Glycans in Fields, which is opposite Imperial Cancer Research Fund. And we spent many summer lunchtimes having our picnics in these fields. So as I said, I went to work with Steve Higgins in Ian Mannering's lab. And we were looking at proteins that were induced in the seminal vesicles, and seminal vesicles are part of the reproduction organs of the male. And what we showed was that actually, if you just look at this top graph, that if you take testosterone away, then the proteins produced by these seminal vesicles drop right down, and when you add it back, it comes right back up again. And although this sort of seems pretty obvious now, in 1978, these were quite novel and new experiments. Steve was a great teacher and he taught me an awful lot, and I really enjoyed being in his lab. But in 1978, I moved to the lab of Professor Joyce Taylor-Papademetrio, and she had recently been recruited to ICRF by the then director, Sir Michael Stoker. Now, Joyce has remained my friend and mentor ever since this time. So she's been a great friend, and I think it's really important that not just women, but really everybody, not only in science, but in all careers pathways, do have a role model and a mentor to look up to. And I've been very lucky to have Joyce. Now, I spent the first 6 or 7 months in Joyce's lab doing what were called radioimmunoassays. So this is an assay to look for antigens in various substances and using radiation to identify that substance. Now, this was because we were looking for human milk proteins, because there had been some reports that human milk proteins could be secreted by breast cancers. And I did hundreds, if probably thousands of assays like this. But in those days, we didn't have little multi-well dishes like this. We had to do everything in individual tubes, nor did we have automatic pipettes like here. In fact, we used to have to use capillary tubing to allocate very small amounts of liquid. So these assays took a very long time. And I'm just telling you this because actually after 6 or 9 months, although the assays worked very well, we actually found nothing, no milk proteins secreted by the breast cancer cell lines that we were looking at. So research is not always positive. It can be pretty negative, and you have to just sort of carry on and just feel that you've always learned something from those negative results. So around this time, Sir Walter Bodmer became director of Imperial Cancer Research Fund, and Joyce worked with a PhD student of Walter's, Jim Arkley, to raise monoclonal antibodies to milk fat globules. So these are globules, fat globules that are found in human milk, and they raised antibodies to these. And I was involved in the first paper here. Now, what are monoclonal antibodies? And I'm just reminding, perhaps letting know anybody that's listening from a school, perhaps they're not quite so familiar. So actually, each antibody producing cell within the body only makes one type of antibody. There's only ever one type of antibody produced by one cell. And so what the Kohler and Milstein, who developed this technique, did was to find a way of isolating these cells from immunized mice and to fuse these cells with tumor cells. And what this meant is that you got a cell line, which was called a hybridoma, that you could keep on propagating in culture for virtually forevermore to make a specific antibody, and these are called monoclonal antibodies. And this was a massive improvement, a massive technique that Kohler and Milstein developed, and they in fact won the Nobel Prize for this, because up till then we could only use antibodies that were made in animals, and this of course would run out over time. So in 1982, I started my PhD and continued working as what was called a senior research officer in those days. Now, I started doing my PhD because at the time, Bill House was actually the assistant director of research services. And he called me into his office one day and he said, had I ever thought about doing a PhD? And I thought about it and I thought, oh, perhaps it would be a good idea, because I really enjoyed lab work. So I went back to speak to Joyce, who was, of course, very supportive. And so I started my PhD in 1982 with my Joyce as my supervisor, but I carried on my work as a senior research officer at that time. Now, part of my PhD was to actually characterize the antibodies that I had spoken to you about in a couple of slides ago that Joyce and Jim Arkley and others raised. So these antibodies, these monoclonal antibodies, were called HMFG1 and HMFG2. And one of the first things that we did during my PhD was to look to see whether these HMFG2 and HMFG1, the antigens that were recognized by these antibodies, could be found in the serum of breast cancer patients. And here we have healthy controls, and here we have advanced breast cancer patients. And you can see that you get a lot more of the antigen that's recognized by HMFG2 and even HMFG1 in the serum of breast cancer patients than you do in controls. Now, this was the first time that this had ever been shown. And it went on to show that that there was a trend that if you had high level of these antibodies to to the antigen recognized by HMFG2, then it was associated with poor survival. Now, we showed this presence of this antigen in the serum for the first time, and others have since then have gone on to exploit it by developing new antibodies to the same antigen and to develop a kit called the CA15.3 kit, which is a kit that is used particularly in the States, but to monitor the progression of breast cancer. Now, I further characterised these two antibodies, looking about how they reacted with this normal milk fat globule that we isolated from healthy human milk, and so-called normal cell lines and so-called breast cancer cell lines. And what we found was very interesting, because we knew that both HMFG1 and HMFG2 recognise the same antigen. So what we found was that when you looked at anything that was so-called normal, like normal milk fat globule or normal cell lines, that HMFG1 always reacted better than HMFG2. However, if you looked at breast cancer cell lines, you had HMFG2 always reacted better than HMFG1. So these antibodies showed a differentiation in their ability to bind their antigen, and yet we knew they were binding to the same antigen. So why was this? So around this time, Sandy Gendler here joined the lab, and as you can see, we obviously made quite a trio here. And I suppose I should say at this time that really I hadn't really encountered much discrimination about as I was a woman, perhaps because I'd worked with a lot of women. Although when I think about it in retrospect, I can think that when I first went to my interview at ICRF, that was my very first job that I had. I was asked, as I had recently got married, would I be having a baby in the next 2 years? And I don't think anybody will get asked that these days, so things have moved on. And the other thing I do remember is that one of the first meetings I went to is I sat next to a middle-aged professor from the States, and he just turned around to me and he asked me, well, whose wife are you? Because the idea that I'd come there as my own self, as a scientist, was sort of impossible for him to think about. Anyway, I think things have moved on from that now. So Sandy actually took the lead in actually cloning the gene, or isolating the gene that was recognized by the two antibodies that I was looking at, MUC1. And we published this paper in 1987, which was the first paper showing that that a mucin can be cloned. Now, a mucin is a very large molecular protein with a lot of sugars on it. And in 1986, I was awarded my PhD, and I went on to look further at characterizing this mucin, and concurrently with publishing the cloning of or finding the gene that actually isolated the gene that coded for MUC1, we were looking at new antibodies too. And this slide here just actually explains to you what a mucin is. So mucins are part of mucus, and everybody I think knows mucus. You know, when you have a cold or when you have a bad cough, you can cough off a lot of mucus. But the main protein or main glycoprotein in mucus are called mucins, and they form this very sort of dense layer over the cells. And this is electron microscopy, so very high magnification of mucins. And in fact, mucins can protect the cells underneath. They can also manage to signal from the outside in, so they can tell the inside of the cell what's going on the outside. And they also have a very important function, particularly in the colon and the intestines, because there seems to be two layers of these mucins. One, a top layer where bacteria, good gut bacteria can live and thrive, and a second layer which actually protects the epithelial cells from any damage and from those bacteria getting down into the cells. So mucins are extremely important glycoproteins. Now, as I said, I was looking at further characterizing the antigen, this MUC1, and what we did was we stripped MUC1 of all of its carbohydrate and raised antibodies to just the core of MUC1. And here, this is what's called SM3, and this is for stripped mucin, and it was the third antibody that we isolated. And if you compare the binding of SM3 to HMFG2, which was one of the first ones that were first isolated, I showed you with Walter Bodmer's PhD student, you'll see that they react quite differently. Now, these are histochemistry sections of the top panels here, breast cancer, and you can see this very brown colour is very strong staining. So you can see both of these antibodies stain very strongly. But the next two panels here and here are benign lesions of the breast. So these panels here that I'm showing are benign fibroadenomas. So these are a benign lesion, they don't form cancer, and you can see there's no real reaction here, there's no brown colour here with SM3, but there is some with HMFG2. This benign lesion down here is a papilloma, and it is very strongly staining with HMFG2. There is lots of brown, but here only a tiny bit of brown. And at the bottom panels here, what you see is a section through a normal, healthy, lactating breast. And what you can see here, you get strong binding with HMFG2, but very little binding. There's very little brown here with SM3. So with a lot of ways to try to see what was going on here. And the first thing we wanted to see was, well, what are these two antibodies reacting with in this mucin? And we isolated exactly the amino acids that this antibody SM3 reacts with, and we found these to be PDTRP. Now this is just 5 amino acids. Now importantly, these amino acids were in the part of the mucin that was heavily covered with a lot of sugar. Now when we went on to look at HMFG2, we found that this was a much smaller epitope, this is called, a much smaller number of amino acids that HMFG2 was recognizing compared to SM3. So HMFG2 is just 3 amino acids, SM3 is just 5. Now what we hypothesized and we went on to show is that what was happening is that when MUC1 is produced by normal, healthy mammary epithelial cells, you get very long side chains of sugars attached to it. However, in the change to malignancy, MUC1 had very short sugar side chains, and some of these were topped off by a particular sugar called sialic acid. Now, why was this important? Because we could actually stain an awful lot of breast cancers with this antibody. So you can use antibodies to stain sections of cancers. And we found, because we found that SM3, which only reacts with this MUC1 when it has these short chains on it, but doesn't react with MUC1 when it has these long branch chains, because the antibody actually is blocked from reaching its long PDTRP epitope. We found that this was really, really common, that around 90% of breast cancers reacted with SM3. So we know that 90% of breast cancers change the sugars that are attached to carbohydrate. Now, that normally happens for some reason. You don't get something happening in the vast majority of cancers without that happening for a reason. And if you just now go back a bit, we want to think really, what are glycans? What are these sugars? So these are sugars that are attached to proteins that stick out here. This is the protein, these are the sugars attached to it. And they, if you look at them under electron microscopy, they form this very sort of fluffy, hairy border. And glycans, or sugars, are very vital for a number of reasons. They are absolutely vital for the protein to correctly fold and to have its correct function. They're really important for cell-cell communications, because cells have to be able to communicate with each other, because a cell has to know it's within a liver or it's within a stomach. It has to be able to communicate. It's absolutely vital for cell environment for communication, because again, a cell has to know where it is within the body. And glycans are really important for cell trafficking. So if you think of immune cells that are traveling around in the blood, when they have sense a pathogen, say in a tissue, they have to get out of the blood and go into the tissue. And it's the glycans that really help those tumor cells, those immune cells getting out of the blood and going into the tissues. And what we've been particularly interested in the last 4 or 5 years is that they're really important in how the body induces an immune response. So we spent a number of years, well, I think years will be the right answer there. Yeah, a number of years actually trying to look at the mechanism to see why you got this change in glycosylation in breast cancer. And one of the things we found is that an enzyme, and it doesn't matter what it's called, but it's called ST3GAL1. So it's an enzyme that has the ability to add that sugar on the end, which I said was a sialic acid earlier. And this enzyme is very upregulated in breast cancers. That means that there's a lot more in breast cancers than there is in the resting breast. And here is just a section, or immunohistochemistry section, and this dark blue area here, this is breast cancer cells. And this area here with the arrow, this is a normal duct within the breast. Now, if you look at B here, what we're seeing is, we're seeing the amount of RNA. So RNA is what will actually, is what's transcribed from the DNA and which will go on to make the protein. And you can see here, and we're looking at the brighter the white color is, the more of it there is. There's an awful lot of RNA being expressed by these breast cancer cells. But if you look at this normal duct, you can see very little expression. There's not any bright white. And if you go down here, this is a section of a normal lactating breast. And again, you can see here, the white is pretty dull. So what we showed was that there was a massive overexpression of this enzyme that could add this small one sugar sialic acid, and that was found a lot more in breast cancers than in the normal lactating breast. And although I don't want you to really take this in, all I want to show you is that what we showed was that in the normal MUC1, you had these long carbohydrate side chains that were attached, and in breast cancer, so MUC1 expressed by breast cancer, we had these shorter side chains that very often finish with these diamonds, which actually is the sialic acid. And this group of sugars here, which when attached to MUC1, makes it called MUC1ST. Now, this particular sugar is not tumor specific. It doesn't it's not found anywhere. It is found other places other than tumours, but what is tumour specific is that this particular sugar, this what we're calling ST, is not normally found on mucin type proteins such as MUC1 when they are expressed by these epithelial cells, which are the cells that do the business within the body. And this is really important. And what's also important, that when they are expressed by the mucins, and this is, for example, MUC1, you can see that there's hundreds of repeats of this sugar. And that makes a lot of difference to how these sugars can interact with receptors on cell surfaces. Now, around this time in the early 90s, Joyce and I started the International Workshop on Carcinoma-Associated Mucins, and this is evolved over time into Mucins in Health and Disease. And this have always, except for one time, and the very first one, been found in Robinson College, Cambridge. Now, these mucin meetings came out of a very early meeting that Joyce attended in San Francisco. And if you look at this picture here in the top left-hand corner, you'll see that there's Joyce. There are no other women here. That was all men. And when Joyce came back from this meeting, she found that she really wanted to get moving on these mucin workshops, because mucin, it was obvious, was so important. And she wanted to keep these meetings within the UK, because in Manchester at the time, there was a lot going on in mucins, but it was more of mucus. It wasn't really clear what the individual mucins were. And from this very small workshop, it has evolved. I think this one picture was taken of the mucin meetings about 4 or 5 years ago. And you can see the participation in it for women in these workshops has hugely increased. Now, we found these meetings extremely important, that Joyce and I now have been co-chair for a number of years, and the last one was in 2019, because it's really put mucin workshops, mucins on the world scene. Mucins were always sort of tucked at the end of other meetings, and people didn't want to know about them. And it's also allowed us, as part of the organisation, our organising committee, to make sure that as many women as we can possibly invite are invited, and that there is a good female population within these mucin meetings. And here, this is me, Joyce, Sandy Gendler, who went back to the States. This is Marlene, who she's from Sweden, and this is Gershon, who's our meeting organizer. But these meetings aren't all science. They're tremendous. They've been so good at having allowing collaborations, having interactions, and getting mucins on the scene and getting us all together. But we do have some fun too, and we always take an afternoon off. And this is the lab going punting, and this is members of my lab, and this is one of our Japanese participants within the meeting. Punting. At this stage, I think we were being punted rather than punting ourselves. And also in the evenings after the conference dinner, we always have a disco, and you'd be absolutely amazed at how these seem to be rather staid scientists can let their hair down at a disco in the evenings. So in 2004, I became a senior group leader at Cancer Research UK. And we were starting to investigate, really, not the mechanisms why we got this change in glycosylation, but what did this change in glycosylation that you saw in cancers compared to normals result in. And these are just some experiments showing that in a spontaneous model of mammary cancer in mice, so these are mice that spontaneously get mammary or breast cancer, if you can engineer those mice so that they express human MUC1 carrying the silyl-T glycan, then these develop tumours a lot quicker than the control mice. Furthermore, if we used a transplantable system of tumour growth, so here we actually transplant very small numbers of tumour cells into mice, you'll see that if you have MUC1 carrying this ST glycan, they grow much quicker than the same tumours that are carrying MUC1, carrying these large branched core, large branched glycans, which are found on the normal MUC1 expressed by cells, normal cells in the mammary gland. However, if we tried these in a mouse strain that's called a nude mouse, and this is because these mice have very limited immune response, then you get exactly the same growth rate. So this made us really think that there was some immune response going on here, that there was something to do with the immune system that was affecting why tumours that carry MUC1, carrying the ST glycan, grew much faster than the same tumour carrying MUC1 but with long branch glycans that look like the normal MUC1. So in 2007, I moved to King's College in London, and I was initially as a reader, and then I was promoted to a professor of glyco-oncology. And you can see here that this is the campus. This is the quad of King's College, which is just tucked behind the Shard, or next to the Shard. So when I moved to King's College, my sort of area of research very much were carrying on looking at the role of aberrant glycosylation in breast cancer. And we've done quite a lot of work, which I'm not going to tell you about, but very much coming through EU grants, looking to see whether we could actually see autoantibodies in breast cancer patients that might be suggestive of the ability or the fact that they were going to go on to develop breast cancers. But we spent a lot of time looking at this, and in fact I'm afraid we could never distinguish between individuals who went on to get breast cancer and those individuals that didn't go on to get breast cancer. So it was a lot of work, but unfortunately it didn't show anything. Although I must say that MUC1 now, as I said, that Sandy and myself and Joyce were the first people to clone mucins, that now is one of the most important or the most worked on antigens in immunotherapy. But when I moved to King's College, I wanted to look really more translational research and to look at what was going on and what really was the role of why were these breast cancers changing the sugars attached to MUC1, and what did that do to the cancer? You know, why was it advantageous? Because so many cancers did it. So we were particularly looking at the role of receptors in induced transcription, trafficking and invasion, and immune modulation. And I just want to mention at this point a couple of postdocs. One is Virginia Ortega, and I want to mention her because she was the major player in the role in receptor. These are how molecules on the surface can induce the induction of genes, and what she found was the glycosylation of the molecules on the surface altered how they could induce different genes. And I want to put her up because actually Virginia is now working at Imperial College, so she's in Tem Faizi's lab, I think over at the Hammersmith campus. And the other person the other postdoc I particularly wanted to mention at this time is Richard Beetson. And Richard did his PhD with me. He then left to go and do a postdoc in Cardiff. He came back to do a postdoc with me. He left again to do a postdoc somewhere else, and now he's come back again to carry out his postdoc, and he's a co-investigator on our MRC grant, and now he's a very senior postdoc, and he's looking to go to become an independent investigator. And he's been looking at how changes in glycosylation can actually modulate the immune response. So I said that this MUC1 carrying this ST, or it's also called sialyl core 1, seems to be able to make tumours grow faster than tumours that are just expressing MUC1 carrying the branched core, branched glycans. And if we look to see, all right, that's fine, but is MUC1-ST really expressed in real life? We know there's a change in glycosylation in breast cancer, but is this particular glycan expressed? And here we have staining of breast cancers here. And at the bottom here, we have staining with MUC1-ST, and this is a very poorly stained one. And this, we're looking at this very strong breast brown stain, a very strongly stained breast cancer. And about 78% of breast cancers actually do express MUC1 carrying this particularly glycans called sialyl core 1 or ST. This is really important, because if real tumours aren't doing it, it's really not important what we see in the test tube if real tumours can't be expressing the glycoprotein, the MUC1 glycoform that we're looking at. And so what Richard found was that MUC1 carrying ST actually binds to a type of immune cells known as monocytes and macrophages. And here this just shows the binding of MUC1-ST to these cells. So these are monocytes and these are macrophages. And what was also found is that you could inhibit the binding of these cells by using an antibody to something called Siglet-9. So if you use an antibody, you block the ability of the MUC1-ST to bind to its binding site. And in here we found that it was actually Siglet-9. So what are monocytes and macrophages? So monocytes circulate in the blood, and when they move into the tissue, they become macrophages. And this is a scanning EM of a macrophage, and you can see it's quite a large cell, and it has the ability to phagocytose, so to eat up pathogens, dead cells, and that type of thing. So it's a way of sort of mopping up all the rubbish that the body doesn't want. But it also has the ability to try to stimulate T cells. Those are the cells that actually can do the killing of cells, like pathogen invaded cells or even cancer cells. And what is this molecule called Siglet-9? So this is something that's found on the surface of immune cells, particularly these monocytes and macrophages, that can actually bind to this sugar called sialic acid, this one that's always been represented by diamonds. And when this binds to sialic acid, it's been thought that it can sort of turn off the immune response or can modulate the immune response. And so this is a very busy slide, and I really don't want you to take in the actual detail of it, but what it shows is that treatment of macrophages with this particular glycoform of MUC1-ST, results in the secretion of factors that are associated with tumour progression. So if MUC1-ST engages macrophages, then it can induce those macrophages to secrete factors that actually induce the tumour to grow more. And this is really important because this is all about the tumour environment or the tumour microenvironment. Now, till, I don't know, 10 or so years ago, all of the focus in looking at cancer was really looking at the actual cancer cells themselves. But over time, this has evolved, and now it's become really important not to just look at the cancer cells, but to look at all of the cells that are surrounding the cancer, because it's become clear that the cancer cells here can actually talk to immune cells or fibroblasts. Those are the cells that sort of make the structure of organs, and that this talk between cancer cells and the immune cells and cancer cells can really affect how a tumour cell can grow. And so on the left here is just a cartoon. On the right here, this is an actual breast cancer, and this is shown in this sort of turquoisey colour here. And you can see around here red cells, and these are immune cells that are surrounding the tumor nest. So this is like called a nest of tumor cells. And then there are also red cells that are actually invading into the tumor nest. And there are a lot, the yellow here is collagen, the way that the tumor is sitting in this collagen. So there is this massive talk between the tumor cells and the cells that are surrounding the tumor. They're not cancerous cells themselves, but they can actually induce the cancer to grow. And so what Richard showed was that monocytes and macrophages, educated by MUC1-ST, via its ability to bind to this protein on monocytes and macrophages called Siglet-9, can secrete factors which induce tumor growth. And again, a busy slide, but I just want to point out here that when you add MUC1, this protein called PD-L1 is upregulated. It goes from here to here. There's more of it on the monocytes and the macrophages. And why this is important is this particular protein is known to engage with its counterpart on T cells, and these are cells that can kill the tumor cells. And when this particular protein, PD-L1, engages with its counterpart on T cells, it turns those T cells off. So what is happening is that MUC1-ST binding to the macrophages is causing the macrophages to turn on molecules that actually then inhibit T cells, the killer cells, doing their job. And this just shows here that factors secreted by these macrophages actually also inhibit the growth of T cells. So coming back to our picture of the tumor microenvironment, it's not only that the tumor cells and the macrophages can talk to each other, but actually the tumor cells can talk to the macrophages, which talk to the T cells, which then can inhibit the immune response. So this massive interconnection of the way cells are talking to each other within the tumor microenvironment. Now, in January 2017, the lab was really lucky enough to move into the new cancer centre at Guy's. So this is mainly an NHS building that is just concerned with cancer. And we and King's College have a floor on the 9th floor, which is here, which is a pure research floor. And it allows us really good interaction with clinicians and researchers within the NHS and with the tumour bank that is located here. So these were the labs when we first moved in. They were absolutely brand new labs. So there was a number of different labs moved into these, and you know, it's really great. And I had when I was full professor and not emeritus, a very nice office on the corner down here. So what Richard showed, and now we're coming up right up to date with some research, is that MUC1 carrying this glycan MUC1-ST can induce the differentiation of monocytes to macrophages. Now, you didn't need anything else, and this is shown in a test tube in the lab. You didn't need anything else if you just added this MUC1-ST to monocytes, which we can isolate from healthy blood, then you could make macrophages. And these and you can just see here, this is MUC1-ST, that you needed this S, you needed the sialic acid, because if you took the sialic acid away, this didn't really work very well. And this is the normal factor that induces monocyte differentiation. Now, these macrophages that have been just induced by MUC1-ST, nothing else, they have the characteristic of these tumor-associated macrophages. And here I'm just talking about this molecule PD-L1 again. This is this protein that has a, when it binds to its a counter protein on T cells, has the ability to stop the T cells from reacting. They just can't recognize antigen anymore, so they can't kill. And MUC1-ST, this upregulates this PD-L1 in tumors. on the macrophages. And this again is too busy to load, but all it's trying to say is red means it's up, green means it's down, and this is looking at a whole range of messenger RNAs that are expressed by these macrophages. And what you can see is when you induce these macrophages by MUC1-ST or by the normal chemical or normal cytokine that's normally used to induce these macrophages, you get quite a different profile of proteins being expressed. So they're quite different macrophages, and this is quite important. They're quite different. But as I said before, if these macrophages aren't in real life, we've been looking at what's happening when we can isolate monocytes from blood and induce them to become macrophages with MUC1-ST in vitro, i.e. in glass, in the test tube. Are they there in real life? That's what's important. And here, these are breast cancers again, and this is stained with MUC1-ST. This is stained with a macrophage marker, and this is looking at the messenger RNA of a particular protein that is characteristic of these MUC1-ST. And you can see that there's a correlation between the macrophages that are induced by MUC1-ST and the presence of MUC1-ST. And here again, we have two factors, and here that actually bind, have two factors that are actually expressed by the macrophages that are induced by MUC1-ST in the lab. And again, we see that these factors, these macrophages are present, and we're looking at these yellow ones here because we see both. So you can just see where the arrows are pointing, these yellow. These macrophages that express the proteins that are characteristic of macrophages induced by MUC1-ST are found around these nest of tumour cells in real life tumours. And what Richard showed was that if you look at tumours, or if you look at patients that have lymph node involvement, so lymph node involvement is always a sign of not such a good prognosis in these breast cancer patients, because the cancer is actually spread to the lymph nodes under the arms. You'll see that there's a less percentage of lymph node involvement when you don't have MUC1-ST than when you do have MUC1-ST. So this looks like MUC1-ST is associated with more lymph node involvement. And again, if you look at a signature of the proteins expressed by these MUC1-ST, if you have high levels of these in tumours, and this is done by analysing public databases of hundreds of thousands actually of breast cancers that have been analysed for the messenger RNA that they're expressed, the patients do poorly if they have high levels than if they have low levels. And this here. Now, I brought you right up to date with our research, so I just want to re-summarise my career pathway. So I started off as a research officer, and during this time I did my PhD and became a principal research officer at Imperial Cancer Research Run. I then actually became a staff scientist, which is a scientific position but really within somebody else's lab. I then became a senior scientist, senior group leader at CRUK, and then transferred into the university sector, became a reader and a professor. So there's a point down here that I'm trying to make, is that 27% of UK professors are female, but 42% of full-time academics are female. So why is this? I mean, if 42% of female academics are female, then one would expect that 42% of professors should be female. It's too long now for people to keep going through the argument that, oh yes, well, in a few years' time they will become, because this has been going on for years now, and it's not just that we're having to work our way up. So why is it? Well, of course, I don't really know why, because if I did, then life would be a lot easier. But there are a few things that perhaps we should think about. That there's only 41 of the top 200 universities in the Times Higher Education World University rankings that are run by women. So obviously women are not really in the top jobs. I think there's a lot of unconscious bias. I think there's a lot of men in the positions where they can actually recommend women for these higher jobs, and perhaps women aren't what's coming directly to their mind. Perhaps they're thinking more of men for these top jobs. But I also think that some women really need to believe in themselves more and put themselves forward, because it is really important, this, because I know plenty of women who have said, well, I could apply for a professorship this year, but I don't think I'll get it, so perhaps I shouldn't do it. And whereas when you speak to men, they say, well, I'm going to apply for it this year. I might not get it, but that doesn't matter, because if I don't get it, I can do it next year. So I think we've got to really entice our women and really incense our women to think more like that. If you fail this year, don't worry, you might get it next year, rather than keep putting it off, thinking, oh, I don't think I'm quite going to make that grade yet. Just try, just give it a go. I think that's really important. And what helps women make a career in science? So I think one of the things is having a supportive background and a supportive bringing up. It's not essential, but it certainly helps, because I had very supportive parents who never made a difference between my sister and I and my brother. We were all expected to do things. Also, I think it's vital, actually absolutely vital, for women, if they have a family, to have a supportive partner who takes their fair share of household duties and childminding. And as I said, I married my husband when I was 20, and we've been happily married ever since. This is him here on a lake in the US. And also I think it's important to have the backing of your children too. And here, this is my son here, and I must say that, you know, when he's 31 now, but when he was small, he never minded, or he never moaned when I went off for a few days, or I went away for a week at a meeting, because his father had always been involved in his child caring, and so it wasn't such a big deal. I also thought it's really important to have, I think, to have a real role model, mentor and friend. And here I want to talk about Joyce again, because Joyce Taylor Papademetri has been a real role model for me and a great friend, which has lasted for 40 years or so. And I think it's vital to have a great lab, and this is a recent picture of the lab. But what does a career in scientific research has? What's it really given me? So I think the most exciting thing is that it's given me the ability to be my own boss and to follow ideas. That is amazing. You can go to work every day and you think, oh, you know, what am I going to do? What can I do? And how can I, you know, follow my idea? And I think it's a really privileged position to be able to do that. I think it's given me the excitement of finding out something new. You know, sometimes these can be quite minor things. It can be just one tiny experiment and you see something really interesting, say on a Western blot or a gel, and you're just really excited by it. It's really, really exciting. I mean, it's nice to feel that your job's worthwhile. I think that's an important factor too. And as an academic in a university, it's absolutely great to see your students blossoming, both your undergraduate students and your postgraduate students. It's given me the ability to interact with a diverse range of nationalities through international meetings and grants, and particularly the grants. We've had grants from the European Union and from the United States in the NIH grants, and this actually has been great. It's given me the ability to try and help others with their career pathways, and I think that's something as women that we really need to do. And it's allowed me interaction with great people and the making of many everlasting friendships. And I just want to finish by thanking everybody who I've ever worked through throughout my career, which actually I have been lucky because I have found very little discrimination that I am obvious, that is obvious to me in my career. And I want a special thanks to all the lab members over time. And this is just a group of us when we were doing the moon marathon in aid of in aid of breast cancer now. And I'd particularly like to thank Joyce, because Joyce has been a great mentor and a great friend throughout my career. And of course, the funders, which really, to carry out a career in research, it's just absolutely impossible without the funders. So thank you very much, and I would now like to end the talk here. Thank you.
Okay, thank you very much, and I'd like to applause your hard work, success, and the great presentation. And thank you very much.
Thank you.
And now we have a few minutes left, so I want you to ask any question if you have, or somebody wrote a question. Hold on. The publishing. What is it? Fantastic talk. Thank you. Really nice story and the progression of research. Thank you for talking about your career also. Okay. It's great. Anyone have any questions, scientific or career, or any inspirations you want to ask? It's a rare chance to ask. I actually want to ask just one question. Joy, you had a quite successful career, obviously. And what do you think? What is the key to your success?
If I can be completely honest, I think a lot of it's luck. I mean, it's quite important, and sometimes it's just luck that you're in the right place at the right time. I mean, I think I was really lucky that I decided right at the beginning to write to the Imperial Cancer Research Fund to work there, because at that time it was just an amazing place to work. You know, David Lane was there. Numerous Nobel Prize winners were there. Molecular biology, I remember, was just starting off when I was there, and we used to have the idea that the 5th floor was just full of macho men doing molecular biology, which is not good for women's research, but that's what's happening. But then I think I had a great mentor as well. I mean, when I went to work for Joy, she was always really supportive of me, and that is really important. But I think it's a dogged determination too. Sometimes you want to put things off because you don't feel you can do them, or you don't feel that it's the right time. And I think sometimes you just have to go for it and do it, because sometimes it's never the right time, and it's never, you almost might think that perhaps I can't do it, but you can. Women have they have a huge talent that really needs to be exploited. And please, go out there, think you can do it, because I'm sure you can.
Okay, thank you. And there are a couple of questions. First is, which remarkable breakthrough in cancer research do you personally think is coming in the near future? So that's probably on top of the checkpoint drug already available now. So what next?
Well, I could be really biased and say that I think glycoimmune checkpoints are now becoming really, really important. And so these are checkpoints that actually inhibit the innate immune response rather than the adaptive immune response. That is what the checkpoint drugs that are in the clinic at the moment are doing. I think the technology now is moving so fast that you're able to sequence the sequence the gene and look at the RNA from each individual cell. And this is going to tell us so much and going to move, I think, science forward really fast. And technology is absolutely important for moving science forward. And, you know, Kohler and Milstein, when he first made those monoclonal antibodies back in 1975, they absolutely revolutionized science, because if you think about drugs like Herceptin, which is a humanized monoclonal antibody, and it has revolutionized the HER2 positive breast cancers in their treatment. So there's a massive way that technology can move things forward. So I think it's technological advances that will be able to move things forward very fast. And I'm hoping that glycoimmune checkpoints, which are now becoming quite really important, will actually be an exciting innovation.
And another question is, well, it's quite similar. Very nice work. I would like to ask about the possibility to use glycosylation to inhibit GAL receptor. For example, GCS100ID galactine 3 antagonist. Do you believe in using glycans as a therapeutic agent against cancer?
Yeah, no, to be truthful, I'm Glycan inhibitors, I find maybe I'll be very tricky to use, because glycosylation is so important. You know, it's needed by so many functions of the body, and so many functions of individual proteins, glycoproteins, that to actually inhibit it, I think, might cause very nasty side effects. And I try to emphasize that this ST glycan that we're particularly interested in on MUC1 actually is not tumor specific in itself. It's tumor specific when it's expressed by MUC1. So if you start to, say, inhibit the enzyme that adds the sialic acid onto the core 1 or the T antigen, you're going to also inhibit it when it's used by normal healthy blood cells, and I think it's going to be very tricky. So I think to use straight glycan inhibitors is going to be difficult, and I think it's going to be too difficult to do. There are ways of manipulating the glycan, particularly on cancer cells, very indirect ways, and that may be more of a way forward.
Okay. So our next question is about career. So if I want to pursue research in cancer, but if I have not done it as an elective in my undergraduate, would that pose as a problem?
I assume the questioner means that they haven't taken cancer as a particular mod. Now, I can't see that as a problem. Personally, I don't see that as a problem at all. I mean, I think nowadays it depends what they want to do, whether they want to go on to be an academic, so they want to go on to do a PhD. Nowadays, it's becoming more and more difficult to get a PhD straight out of undergraduate, and perhaps doing getting some experience in a lab or doing a master's is a good idea. Because I just know for some of my students who have been excellent undergraduate students have had real trouble getting PhDs without having gone on to do something else other than just straight out of their undergraduate.
Okay, so 2 more questions. One is from Professor Carl Drickmar, asking, saying first, you define a new form of active retirement. As one doing basic science with clinical impact, does the NHS University hookup really work?
It has done in the cancer centre, because we have in the cancer centre we have a very large tissue bank, and actually tissue banks are really, really important, because you need to have tissue stored down, because if you find anything, you need to be able to go back to look at that tissue. Now that is funded through King's, but it also has a big NHS funded, and it actually is working extremely well. Within the Cancer Centre too, a number of the PIs who are doing academic research are actually active clinicians. So that, you know, having to be able to go downstairs to look at your patients rather than going across London or makes life a lot easier to be active academic research clinicians. I'm not a clinician, but you know, active academic research clinicians and acting clinicians, consultants at the same time. So within the Cancer Centre, we have a number of PIs that do that, and I think that's working very well.
Okay, thank you. So 2 more questions. So first is, did you ever feel like you had to choose between your career and your family?
Yes, I think I did actually, because I think perhaps very early on in my career, I would have gone abroad to do a postdoc, but I decided not to because that was family commitments and I didn't want to do it. So there are times, yeah, that you do have to. Yeah.
Okay, so last question is, great talk, Joy. If you could dream, what technological breakthrough you think could push glycobiology forward? So what kind of new technology?
I think some technology that can assay glycans really fast. I mean, that's what was needed. You know, it needs because they're so difficult, because they have not template. You know, we all know they're not template driven. They can be anything. You know, it's so easy now to look at DNA, to look at RNA, and to say what the protein is. You just can't do that with glycans. And although the technology has moved tremendously with glycans, and I know at Imperial, obviously with Andel and Stuart Haslam, that has been a huge move forward. It's still quite time consuming and difficult. And if there was some way of really being able to tell what glycans were there quickly, I think that would be amazing.
Okay. So actually, 1 more question came, and this is also technology and for the future. What do you think about using artificial intelligence and machine learning tools to facilitate the processing of the data? Would it be useful or not?
This is slightly outside. AI is slightly outside of my expertise, but I just would like to say that I know that there are a number of groups within King's. Our school is led by Tony within King's, and I know he's very much working with AI people to look at way to analyze data, to analyze imaging data, to be able to put data together from not just imaging, but from say serum assays, imaging, different types of imaging, to put those together and use the AI to actually identify factors, or to identify a signature that could be associated with early diagnosis or bad prognosis. So I think AI definitely has a role. I'm not sure at the moment, and this is actually not my expertise, but I do see from what I see from others, it will start to play a role as we go forward.
Okay, thank you very much.
Thank you.
And I think if no more questions, then I really would like to thank Joy once again. Thanks for your great presentation and sharing your story, and we really do appreciate. And that's really fit to our Department of Life Science International Women's Day lecture theme, and that will be great. And those of you who couldn't join from really beginning or so, we will put this video onto our website, so please have a look there if you're interested. And thank you very much, Joy. So
thank you very much. Thank you.
Thank you.
Thank you. Bye-bye.
Bye.
Thank you everybody for coming.